And so when we look at our kidneys, we have to use our imaginations and remember that this renal pyramid that we're seeing here is going to be, again, in our imagination, sort of a filter, right? Something that's catching what will be eventually urine. And all of them are pooling that what will be urine into these little funnel pathways, these little duct systems here. And this is called your minor calyx. And if we draw into our minor calyx, let me make this a little larger, into our minor calyx, it's going to be that space that we can see that is underneath our renal pyramids. Right underneath. It's where we're urine and we're traveling down these pyramids. We're going to go, our first stop is going to be that major calyx. And then you can see we have another term called major calyx, and that's going to be everything after the minors where they start to union, right? Where they form a union, in other words, here. So here's another major calyx, and in blue here we're seeing our minor calyx, and in purple we are seeing our major calyx. All right, so again, this is just, in our imaginations, just a pathway. Like if you're looking at water draining down a pipe, a sewer, or anything, they're just going down, and there's names to that system. The first part is the minor calyx. The larger part where they all combine is going to be the major calyx. And so you can see that multiple combine here, major calyx. And then finally we arrive at our renal pelvis. Our renal pelvis is going to be where all the major calyces unite. In other words, where all these purple ones, all the major funnels, these large ones will finally combine and then finally pool their urine into this renal pelvis space. That renal pelvis space will send urine down the ureter. And we might as well just quickly finish our review there. Keep going on our review here. That renal pelvis will lead to the ureter. Try to remember that this whole structure is your kidney. And this is going to be another associated structure of the urinary system, which sends urine or allows urine to travel from the kidneys. And again, this is the ureter to arrive at our urinary bladder. And this is where we're going to store urine. So you can see how our urinary system is a little, the parts are sort of separated and only really connected by this ureter. And this ureter will serve as a muscular straw, as we're going to see a little bit later down the line. And with that, other topics or other things we really want to be well versed on before we move on to more complicated topics is this region that we're seeing that I'm highlighting in my red right now. This is referred to as the medulla. So this segment here, medulla, everything where your renal pyramids are is considered the medulla. And you can sort of see that there's a little bit of a segment left over, and I don't want to lift off my pen because I'm going to lose the red here. But if you look just at the edges of my red marker lining, I would say like around here, you're seeing these edges. If you go just to these ends, you'll see that there's always a little bit extra left. And so everything in that space is called the cortex. In other words, I'm going to highlight our cortex now. And so this is going to be our cortex. And so here you can see our cortex. So that's going to be the space that's on the outside of the medulla. And so the word cortex means area outside, area outside. And medulla means area inside, inside. All right, everything inside. And with that, some other quick terms to refresh your memory. If these are pyramids, renal pyramids, then these right here are going to be renal columns. You can see that there's a bracket to indicate that very thing here. And so the medulla is made out of nothing but renal columns and renal pyramids when you really think about it, right? Because if we wrap around I'm just going to do this part of the medulla, this little portion here, then you can see it's taking up pyramid, column, pyramid, column, pyramid, column. So the medulla is made out of renal pyramids and renal columns. And so you can see that's why I have this little note. Renal pyramids, again, lead to renal pyramids and columns. And renal cortex, sometimes you'll hear the adjective cortical, which is just an adjective of the word to describe that it's in the cortex. And so for easy learning or application, just think of the word as area, or even better, outer area for now. And as we move forward, last thing is there's also little tiny segments to the kidney. So for example, there are lobes to the kidney. And so the kidney is going to comprise of lobes made up of half of the columns, the whole pyramid. You can see that's another lobe. I'm going to draw another one here. Here's another lobe. And then here's another lobe. So this little dashed line is here to represent that for you. And so a kidney lobe is just those little portions that what you see in the dashed line. A little bit of renal column, a little bit of renal pyramid, just to elaborate on our terms. Make sure that you can recognize this image. All right. And in lab, you will have to learn these terms as well. So keep that in mind that you're learning lab material for the last lab exam, which is approximately 3 weeks away, and your other lecture exam. And so it's all tied very closely together. So that's why I'm sort of going over this gross anatomy again, because we won't have time to go over it in lab again, but we will have time to just, take out the models and stay organized and allocate some time to each thing. And so with that. And so we left off on our blood vessels, and just quickly, another refresher here. We had the unit inside of the kidneys, which is going to be doing all the filtration. It is filtering what is the contents of your plasma, and it's going to select for things that should stay back in the body, or that's it. It can actually do that. It can select that something needs to go or something needs to stay. So they are little, you can think of them as little trash selectors. They're cleaning out your plasma of waste that your cells produced, all your tissues. And so there are 2 types of nephrons. There's a cortical nephron, which again means to reside in the cortical. And then we have juxtamedullary nephron, which means to be next to the medulla. Next to the medulla. If we look at our regions here, renal cortex is going to be this outer area. And then we're going to have this pinker area on my screen. I know yours doesn't have color, but the renal medulla. And so, again, we are cortex and medulla. Okay? So we are seeing that dividing line of a pyramid right here. And so if it's next to the medulla, next to medulla. That's what juxta implies, next to the medulla. And so if you look at the differences between 2 cortical nephrons are the most abundant, being at 85%, located at the cortex, and they are doing most of the regulatory functions, such as regulating blood volume, regulating or keeping track of blood pressure, even keeping track of salt levels. It can do a lot of those things that you're going to learn in physio. And then the last 50% are juxtamedullary. And one thing to add about this is what is it to concentrate something, really, when you think about it? To concentrate something means to make it stronger, to make it more, to be more of something without water. In other words, you're sort of removing water in order to make it very, very more of your solute. And so let's just draw this out. If you have, let me draw it with a marker. If you have a cup of something, a solution, and then you place a certain amount of solutes. I'm just going to write 10 little molecules. This is my molecule right here, 10 of those little molecules. And so if you compare it, if you want to make it more concentrated, what you do is that you sort of remove the water level without removing the molecules. So now there are more molecules occupying a smaller space, and so we consider that being more concentrated. If you wanted to sort of understand, or we all have a general understanding, if I give you a cup of water, bless you, if I give you a cup of water, and then I put a little bit of tapatio in there, a little bit of hot sauce, right? 10 drops of hot sauce, and the cup is full, you won't really taste the tapatio as much. I mean, you might taste it, but what happens if I add those same 10 drops to this one with less water? And then so this one will be more concentrated, and you're really going to taste that tapatio or that hot sauce when you drink just that little bit of water with just those 10 drops. And so we consider that to be more concentrated. And so this segment here is going to be regulating pretty much how much water reabsorption is occurring. And this is something that physio is really going to grill you on. I only really care for you to know what is considered a juxtamedullary nephron and to know the percentage compared to the cortical nephrons and their location, and that they concentrate urine. But this is a fun fact for physio, that this is going to be a portion where osmolarity, or a lot of the movement, liquid movement, occurs between those tubules. if we look at these little nephrons now, we always consider this as our starting point. And so this is going to be, when we think about these little blood vessels, they're going in and feeding blood. So this next page is about understanding blood flow into the actual cortical nephrons, which are again located, let me get it on the screen here, located on the cortex. And then we have some that are juxtamedullary. And so blood, if we look at these red ones, they're responsible for delivering the plasma that needs to be filtered. And then once it's done so, right, it goes into those nephrons, we can all expect that the veins bring out everything that survived the selection process and decided to not be urine. So again, red is bringing in whatever will not be removed as waste, will return to the vein, and then the vein will take it out of the kidney itself. And so this is a nice little labeling exercise. I would practice labeling here, but one thing I really love this visual because you can see a cortical nephron and a juxtamedullary nephron, and you can see how the blood leads, and then they're all going to have an associated nephron. So the kidney itself, when you look at it, it's made up but of nothing but these little disposers of waste out of your plasma, the nephrons. And you just have to use your imagination that there's one here, and then all their tubes, they look just like it. So they have an extensive tube network. And so what you end up with is nothing but a bunch of convoluted tubules. And that's going to be a vocab word in a second. And it's written there for you somewhere. I'll get it there when we get there. And so here you can see convoluted tubules. So when we look at our microscope slide here, sorry, I can't find my microscope slide. Oh, other page. And so when you look at our microscope slide, this is what you would see on a microscope. This is that same nephron. You have an artery running through here. You can't really make it out in this case, but you can see all these tubes around it. And you've all experienced these tubes. This is an example of simple cuboidal. You're seeing one cuboidal cell here, one cuboidal cell here, another cuboidal cell here, and so you are seeing cuboidal cells. And what were cuboidal cells? What was their function, their specialty? In glands, but why were they good for glands? What do glands do? Secretion and absorption. And doesn't that make sense that this tubule network is going to be made out of simple cuboidal, I lost myself. Where's that nice one? Oh, here. It's going to be made out of this type of tube, made out of cuboidal cells. So things are traversing here through the lumen, or the empty space on the inside. And then so as it's going through, it's being selected for, right? These cells are like, no, wait, I can use you, so come back in. And then they place it back into the capillaries. But everything else just keeps traveling through that tubule. And so with that. And so speaking of blood flow, back to blood flow, looking at just the arteries, they're not very hard to learn. Renal artery is going to be the one that leads into the kidneys. And then it's going to break apart. We saw that we had the segmental branches when we were discussing the lungs. And so this is the same thing. Segmental means to branch, to break apart. And so you have your segmental arteries where we begin to see the area of branching. You can see that there's 1, 2, 3, and each one wants to serve a segment. Remember what this segment, if we have a little bit of a renal column and a little bit of a renal pyramid, what do we call this whole portion? Starts with an L. A lobe. So this is our renal lobe. And so it helps remember the next one, interlobar arteries, because there's arteries running within that lobe. Then we have our arcuate arteries that if we look at our pyramids, they wrap around, and once they're on that horizontal segment, then they become our arcuate arteries. This next word, cortical radiate arteries. This is going to be all the ones that go to the cortex, cortical, right? Let that cortical, you have to log that term away, cortical. It means in the cortex. Radiate means to spread across, to radiate, to spread across. I'm going to put spread across because I don't think that's the perfect translation to that. But otherwise, cortex and spread across, and that's it. So you can see that these are the cortical radiate arteries because they spread throughout that cortical area, and each one will have a nephron associated with it. With that in mind, there is an artery that must go in to that nephron. And there's an artery that will lead out. And so the one that leads into that nephron is going to be our afferent arterial. And I'm going to make that note here. Into nephron. Afferent, into nephron. And our glomerulus is going to be what's doing the filtering. And so we might as well discuss that with this photo. So here is our nephron. In other words, we are zooming in on these little cortical radiates, and we're seeing now how blood is really going to be filtered, just the basics right now. And so if we zoom in on those, this is what you would see on a complete nephron. And if we zoom in even more, this is what we would see. And what your eyes should start training themselves to see is that you should see this blood vessel that will lead in. So this one will be our afferent arterial. I would write that down, our afferent arterial. And the little arrows denoting that for you here. You can sort of use that arrow. There's another arrow out here leading out. And so if it's leading out, it's considered efferent. Let me write these down for you. Efferent and afferent. Oh, dang it. Efferent. And so you can see that this came from the previous artery, which was the cortical radiate artery. And so right after that cortical radiate artery, blood or blood plasma, they're all going to form ROLU and go in one line, all the red blood cells. They're going to travel through and they're going to exit out. But along the way, some things are going to be selected for as waste. And so water can go through here. Water won't necessarily be a waste, but water does get through. And then other things that your plasma, that we excrete as waste in our plasma, such as urea, and so ammonia as well, those things, right, they need to be removed, and that's why urine is characterized as having urea, having ammonia, and so it has to be selected for, and then it's going to go into our first duct system, and that first duct system is going to be our proximal convoluted tubule. Proximal convoluted tubule. That's going to be our first path if we become filtrate, right? Something that needs to be further selected for. You could even see the microvilli are already there on their cuboidal cells, ready to start absorbing or secreting anything they might need to. And the next thing to add is that you might see these little spidery, these little octopusy cells looking here, like octopus cells. And so these are our podocytes. Podocytes, named after, it's like translated over, it means like foot cells. But otherwise, podocytes. Podocytes. And they're right on top of these capillaries. These capillaries are exactly the same little tube network that you learned that exchanges oxygen when blood is traversing through capillaries. These are capillaries, everything underneath these podocytes. So you can see that this is a very unique situation. And they are also helping with the filtration process and the selection process. And so still following our blood path, because that's really the goal right now, is just not really losing track that we're still keeping track of how plasma comes into the nephron. And so we're seeing now how plasma entered this nephron, got selected for, and how there's some structures we learned about. And then our blood cells will travel out of this efferent arterial, and so will the plasma. Everything that was not going to be considered waste. And so with that, the structure around this capillary, right before we finish discussing where exactly does the blood go? Where does the plasma go? Because that's really the last little bit we need to add is how does the plasma get out now? And so what it went into, however, is these podocytes and this blood vessel system, these little capillary systems, are considered a glomerulus. Glomerulus. our glomerulus. And these capillaries are an example of fenestrated capillaries, capillaries that have tiny pores that allow substances to go through them. And then also these podocytes are also selecting things between their little extensions that are on their legs. And that's the role of podocytes. Altogether, this entire thing is called the renal corpuscle. The space inside where the plasma would move over or actually go through the glomerulus is going to be considered your capsular space, or the glomerular capsule. The renal capsule is more appropriate. And then lastly, tubule now. This word, we have to adapt to it, that really we're always saying, and I'll just even write this just to make it small tube, and it just happens to be small, tubule. Like it's the word tube, but a miniature tube. Don't need to memorize these kind of facts, but you should know that that's where the filtrate, everything that's going to be selected for as filtrate, will go through our tubule, which again, the first one we encounter is proximal convoluted tubule. And here are labels for renal corpuscle in case you want to get them now. Renal corpuscle, again, it's going to be the outer capsule. Glomerulus is going to be the capillaries. And all right. So I'm trying to go slow so I don't lose you on this part here. And now when we think about the return back, we travel out of our efferent arteriole. So what we got to add here is we got to add one more thing on our notes. Oh my goodness. All right. So if we look at just our glomerulus in our renal corpuscle, you can even see that first bit of that tube network. This was our afferent going in, and now we have finally made our way to the efferent arteriole, and this is where it gets complicated, and hopefully I'm driving home a very important topic. We know that this tubule can do two things. It can passage urine, and it can also select for things to return back into circulation. So it's not as simple as efferent arteriole going straight into cortical radiate veins. It's actually one more step in between. We know that plasma is going to exit the efferent arteriole, and there's going to be capillaries now called peritubular, which means around the tubes, capillaries. So all of these tubes have extensive capillaries around them, so that in the case that this tubule decides, hey, no, this is water, I need this water back, it can take it out of the tubule and place it into these capillaries, and then we can return back into circulation. So not only does the plasma that was, passed the filtrate test, that passed the test that, the podocytes and the glomerulus said, this should keep going, it's okay. They will also keep going through these capillaries, and along the way, they might come back to meet things that were absorbed originally, but then they get placed back into those capillaries. And so everything in those capillaries will flow right out into what you can logically know as your cortical radiate vein, which is again going to be just the same thing, everything found on the cortex, the ones that radiate out of the nephron. Sorry, this thing's going crazy on me. And then we have the arcuate veins. The arcuate veins are the same ones as the arcuate arteries. They're the ones that run, on the base of the pyramids, arcuate arteries. Interlobar, in between the lobes. And there's no segmental, so finally, if it's going into the kidney, the renal vein. And that's the full story. So here what we should add, we should even just write peritubular capillaries. All right. And so with that, I think we are done with blood flow, which is good news. And the rest of it is not so hard. Nice. Okay. So the next thing we need to add is this slide. Now we have to examine what's going on within our tubules. And we're going to keep it very light. And so here for our renal corpuscle, this is going to be, again, what we know as that capsule, right? The renal capsule. And it's going to surround the capillaries, which you can see here, which have these leg cells called podocytes right on top. Right on top. And so this is where all the filtration is going to occur. And on our model, it's going to be here, number 1. So you can see how these boxes are supposed to match with their corresponding box. And so this is what it looks like inside if you evaluate the tissues. And if we go to our first tube network, our proximal convoluted tubule, this is where we start to reabsorb nutrients from the filtrate if our body needs them. And we don't really call it a filtrate anymore. We change it, once the fluid's actually within the tube network, the tubule system, it's considered tubular fluid. Tubular fluid. It's too early to call it urine, right? Because what urine is what comes out of you, and this hasn't come out of you yet. It's still in the selection process before finalizing what will come out in urine. Then we have our nephron loop. And so the nephron loop is going to be associated with water reabsorption. That's the main fact we need to draw from here. The nephron loop is involved in water reabsorption. It's comprised of 2 segments, a thin segment and a thick segment. And so if we examine that now and we look at our loops here, first we started with our proximal convoluted tubule that was next to the nephron. And then you'll see that at some point we are going to go down, 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 down. And so this is referred to as the descending limb because it's going downwards, descending limb. And then we're going to see that when we go upwards, back upwards towards the direction of our nephrons, then it becomes the ascending. And if we look at each segment, right, like, because you can tell that they're clearly different in size, in diameter. If we look at the tissues, you can see that they're made out of 2 different epithelial cells. This is going to be made out of cuboidal cells, the thick one. And then our thin one, what does that look like to all of us? Simple squamous. So that's why it's thinner, because the epithelium is made out of simple squamous. And so that's the major difference. And the reason is, like, if you're wondering, like, why is that? It's because different things are being absorbed here and in the thick segments, right? Because simple squamous is good at small solutes, gases, and thick limbs, you might need specialized cells to reabsorb particular types of ions or anything else. You just might need very specialized cells. And so that's why you'll see differences in the 2. So you can see that this selection process of the filtrate of the tubular fluid is very, very picky. And the body will adjust using the kidneys to draw water back in if it needs it, to send out extra solutes or salts like ions, like anything you've seen, like salt, magnesium, calcium, any of those extra stuff. For example, everyone always takes their multivitamins, right? You're always taking multivitamins. Did you know multivitamins is you only ever really absorb vitamins when you're in a deficiency. So you're really just peeing out all those vitamins, because there's even been like studies showing that they're like wearing out like pipes in housing, like in just our public areas where there's a lot of traffic, human traffic. There'll always be the degrading pipes, and they're blaming vitamins because everyone's taking so many vitamins. But you only really absorb vitamins when you're in a deficiency. And also, just like vitamin C, how everyone takes that emergency after crazy days and they just don't want to get sick. So that's another very big scam, because your body will only absorb anything if it's in a deficiency. And those things have way too much, unnecessary amounts. And so, you can see how this is a very picky process, right? It doesn't care what your health goals are. If you take those vitamins, if it says, oh, it has too much, you have too much already, you have all of this already. You have all of these already. I'm getting rid of it. And then they will just remove it. So it's just capable of removing different things at different segments. And this is what makes The renal physiology, incredibly difficult. So, but if you can hold on to that theme, that this is not just some dead little straw like a Starbucks straw. It is a thinking tube, right? It is processing what is going through it, and then being commanded by your brain to reabsorb, to send out anything that you might want to keep. And so, hopefully, you remember that little tip when you go forward. And so, last we have our distal convoluted tubule. And this is going to adjust the composition by reabsorption or of secretion. So in other words, it just absorbs or secretes. We do not need to worry what it absorbs or secretes. I will let physiology traumatize you. Yes. All right. And now this segment here. You'll notice that there's this final segment in a different color on my screen here, in yellow on my screen. I know yours I don't have color. But you'll notice that it is considered the collecting duct. This collecting duct is going to be collecting all the all the tubular fluid from the other nephrons. So you have one nephron guiding tubular fluid into this collecting duct. You would have another one over here, another one over here. So at the end of the day, this just becomes one major pipeway for tubular fluid from multiple nephrons, whether it's cortical, whether it's juxtamedullary, but they all combine together. And so this collecting duct will finally arrive to the final portion of that duct called the papillary duct. And this papillary duct is our final step where we will begin to eliminate that urine. And so, if we focus on just our collecting duct and our papillary duct, what you have to envision when you use your imagination is that you have a multitude of these inside of your renal pyramids. Your renal pyramids consist of nephron descending and ascending loops, the thin segment and the thick segments, and a bunch of collecting tubes. So in this segment, everything that you see in the pyramid is nothing but collecting ducts and nephron loops. And so if we keep using our imagination here, we know collecting ducts now. All of the nephrons above are putting their tubular fluid in the collecting duct, and as it's traversing downwards, then the final portion is going to be called the papilla. This final portion of the medulla is typically called the papilla. And to remember this, use this image here. Here, the papillary duct. So you can see that this is, I'm going to write here, next is going to be the minor calyx. That first little catcher part for urine. All right, so once we've gone through the papillary duct, again, they are all going to be in this little segment of all the pyramids, the papilla, and urine will come down finally into what is known as our minor calyx. Minor calyx. All right. Anything else to add here? And so in our collecting duct and our nephron loop, something that allows for water to move across is going to be salt concentrations. Salt concentrations determine in what direction water will diffuse through, and that's typically referred to as osmosis when we're talking about the diffusion of water. And so what do we need to know about our collecting duct is that as it's pooling all that tubular fluid, what will be eliminated as urine, it has a bunch of cells called intercalated discs, intercalated cells, and they're going to be absorbing hydrogen and bicarbonate ions. And what you want to write here about bicarbonate ions, regulate pH, and there's a few things, but we're going to leave it there. Regulates pH. So if we know that bicarbonate regulates pH, you've just learned a method in which the kidneys can actually make you your blood levels less acidic. That's just what I'm going to leave it at. So you can see if this salt can regulate pH, this is one of the functions we attributed to the urinary system as a whole. And so bicarbonate itself is considered sort of a buffer, something that doesn't allow pH changes to happen rapidly. And so that's what bicarbonate ions do. And so you can see this collecting duct is still in that process of collecting any fluid and also reabsorption or secretion of bicarbonate to regulate pH as necessary. And then we have our little principal cells, which do the reabsorption of water, and they secrete potassium, our principal cells. Ask away. So in one of these, which one? Oh, that's a little strange. I don't know why they just didn't draw it in there. Or just didn't, I don't know why. If it's a drawing, they should have just included it, but I don't know. Maybe they just didn't notice. All right. And anything else? I guess that's really it. All right. And so, now this next segment's going to be pretty easy to understand because we already know half of it. If we are capillaries, right, we said that if we are the capillary network associated with the convoluted tubules, then we are considered peritubular capillaries. These are going to be the capillaries that surround the tubules. If we are vasorecta, we are the same exact thing. That we are a capillary network, but the only difference is our location. Our location is in the medulla. This segment down here is the medulla, because remember that renal pyramids are made up of collecting ducts and of nephron loops. And so we have the capillary system around these tubules is called vasorecta. So it's similar to the word peritubular capillaries, but it does not have the same name. It's called vasorecta. But all it does, again, we just learned that the nephron loop has simple squamous, it has cuboidal cells, so it absorbs things. And so when it absorbs it, it places it into the vasorecta instead of placing it into the peritubular capillaries. And so hopefully we all understood that. And so moving forward, don't forget, you have cortical nephrons and juxtamedullary nephrons, and this is just a nice little reminder of the capillary networks or have a different name when they're associated with your nephron loops. And then lastly, we have some little regulatories, and I love this image because here they removed a few things. You can see here, this is our afferent arteriole. And then this segment here is where all the rolu are going through, right? These are the red blood cells, and then the plasma is floating through it. And then we can see our podocytes. We can see our renal capsule. We can see our proximal convoluted tubule and our efferent arteriole. And when we look very closely, you'll see that there's another population of cells. The first one is macula densa cells. The next one's going to be extraglomeruli mesangial cells, which are these located here. And you can even see a little bit of something related, right? These are extraglomerular mesangial cells, extra being outside. These are on the outside of the glomerulus. If we're inside the glomerulus, we remove that word extra, and then we are now inside the glomerulus. And then lastly, we have our granular cells. So these cells aren't really discussed as we're talking about plasma flow and tubular fluid because they have more regulatory functions. And so, for example, macula densa detects your salt levels. Everything that's going through the plasma, it detects salt. This is table salt, like what you put on your food to eat, salt levels. And so it's considered a chemoreceptor because it detects chemicals. I'm going to put actually molecules. It's more appropriate. Our granular cells, these are going to be producing something called renin, a hormone called renin. and we'll discuss this very soon. And they're also considered mechanical receptors, which means that they detect force. Let's do friction. No, let's do what's an easy way to remember mechanics? They just get detected or stimulated by movement. In this case, we're going to put pressure because we are talking about blood pressure. Dang it. Blood pressure. And then our extraglomerular cells, these are going to, actually, we really don't know. I know that there's a little bit of information for this, but there's actually, there's just new science coming out on this because we're actually not quite sure what these cells fully do just yet. And so that's why I have this may pass signals between the cells that the population of cells that are there, macula densa and granular cells. Yes, and they believe that because they are interconnected by gap junctions. And remember that our intercalated discs were pretty much connections between cardiac muscle cells, and gap junctions allow chemicals communication between cell to cell, and that's why it's believed that this is what it does. And now to finish up our urinary bladder. Our urinary bladder consists of this little pouch which stores urine, and this pouch is going to be, or the exit for the urine that's stored within here is going to be different between males and females. So in this segment, on this side, we are looking at females. And at this side, we are looking at males. There is this structure here called, you can see a little triangle here, and it's made out of 3 openings. And it is referred to as the trigon. And it is made out of the ureter opening. The ureter opening, again, and these are your ureters traveling up top here. Let's lay some labels on those. And the last opening is going to be this one, and we'll discuss it in a second. And same thing in females. Here's the 3 points of opening. And again, this is where urine has now gone down the ureter, and it will enter or pool into the bladder through those ureters. Now, what is going on in this ureters? That's really what this slide is about. This here is a microscopic image of a ureter. And this ureter is going to have layers of tissue, which you are already familiar with. This particular type of tissue is capable of transitioning in shape. So what do we think it's called? Transitional epithelium. And then there's going to be this lining right underneath that epithelial cells, or right underneath the, let me get a larger highlighter here. And right, this lining here, it's going to be a little thicker in this image here. But what is this lining supposed to be? Basement membrane. And so one word that we're going to have to slowly apply as we progress into digestive or all of these tissues is you really want to consider when you hear the word lamina propria, you are thinking to yourself, basement membrane. And we'll elaborate on that a little bit further down. This next segment is going to be something that causes a movement inside of that ureter. Which type of muscle do you think it's going to be? Skeletal, cardiac, or smooth muscle? Smooth muscle. So we're just going to write smooth muscle for now. And I would even write muscle layer. This will matter later. And something I want you also to apply moving forward is, you know, typically most things that we're going to see in the moving forward in this class, that when we look at substances on this surface everything that's, because most surfaces have mucus on them, they're typically, everything that's the part of the epithelial cells and this lining is always referred to as mucosa. So anything, whenever we're really talking about the epithelial part of tissue, we're really referring to as mucosa. And this is just a little light preview of what's coming on Thursday, so don't worry too much about it right now. And then lastly, we have our adventitia. This is just a layer of connective tissue on the outside that keeps it anchored. It keeps the ureter anchored. In other words, it keeps these structures completely anchored to the body cavity. All right. And so now we've made it into the urinary bladder. And as we look at our urinary bladder, you also find within the inner lining here in this segment, you also find transitional epithelia. Here's, if you draw some boundaries here, this is roughly where our basement membrane is. And so this is going to be our transitional epithelia. And this down here is going to be connective tissue for now. And again, mucosa. Mucosa. And we'll leave it at that for now. All right. And then the last thing to add, there's 3 more terms to add here. We have here we send out urine through the urethra. So I'm going to bracket this out, bracket this pathway, urethra. In females, it's a little smaller, urethra. And now, openings. So in this segment here, you have what we refer to as an internal urethral sphincter. Internal. Just write it out. Internal. And here on the outside, there is an external urethral sphincter. And this internal one is made out of smooth muscle, so you cannot control it. That's why when you need to go, it's slightly overwhelming, right, sometimes? But the external urethral sphincter is actually skeletal muscle. And this is the one that we have to be thankful for when we are in class and it's not over yet and you gotta hold it in, all right? Skeletal muscle. And for females, here, internal. And external. And the same rules apply. Smooth muscle and, skeletal muscle. And let's keep moving forward, and I think that's it. And here are some practice multiple choice questions for you. And so what I kind of want you all to do is